party mode.
Earlier today on one of the interactions, they were asking if Hercules ever just goes down to listen to the sounds, acoustics. And I couldn't answer that. I didn't know if it's ever used for that.
Trevor, do we have acoustic listeners? There's a word for it. Those are, yeah, hydrophones.
We do not have a hydrophone. If you had a hydrophone on Herc, it would sound like all the time.
Oh. Yeah, that's fair.
The hydraulic motor is very, very loud.
Yeah.
Good. I took a guess and said no.
Are you coming down?
No? Are you coming up?
Okay.
Might just be the heaving, but the delta has decreased. I can head up if you want.
No, that's fine.
Okay.
It's all good.
Man, look at all that blue.
There are scientists aboard the Nautilus, but also at the, pardon me, Inner Space Center at the University of Rhode Island, who are also involved in this research.
Do you ever put sensors on Argus?
Like scientist auxiliary sensors?
Yeah.
Yeah, we had a mapper on for a couple dives or one dive this cruise. It's rarer, but yeah, we do.
We're aboard the Nautilus off of the northern coast of California and Oregon at Gorda Ridge, researching the hydrothermal vents here at the base of the Juan de Fuca plate. Presently, they just finished mapping the area, and on the previous days, they collected samples from various hydrothermal vents that Woods Hole Oceanographic Institute scientists will be studying the geological composition and microbiological composition as well at those areas. And, pardon me, this is part of NASA's subsea research. NOAA and Ocean Exploration Trust. I encourage you to go to nautiluslive.org and investigate the different tabs there, fascinating videos, of various expeditions, organisms, archaeology, geology, ROVs, and extensive assortment of topics to investigate. Dr. Marsh,
Yes, Susan.
I would like to know, is it a school of fish or a shoal of fish, or are they different meanings?
I'm not entirely sure, if I'm honest. A school or shoal? I have no idea. Shoal.
Does anyone know?
Anyone? Yeah, anyone. Anyone. I'm gonna skate.
I think a shoal is, to me, it has a connotation of more of a
It's a behavior,
temporary
isn't it?
aggregation. Yeah.
Yeah.
Exactly. Like a shoaling behavior would be them aggregating, whereas a school is like a unit of fish, like a population that in their life history are moving and foraging together.
Makes sense.
I can read you the Google definition. It says,
Do it.
any group of fish that stays together for social reasons is said to be shoaling. And if the shoal is swimming in the same direction together, it is schooling.
Oh.
Nice work.
So it's
Great.
directional, yeah.
No, mine's wrong.
Oh.
I thought yours sounded the same.
Had that inverted then.
So we're schooling right now, I guess.
Touche.
I was curious because I was wondering if it was a British term, you know, that they said it differently than we did. So they actually have a different meaning. Thank you. I think we all learned
Do
something
you think
new.
a lot of scientific terms change based off of your geographic location? Versus,
Probably
like
not.
America and Europe.
Pretty sure that's like why they use Latin terms so much is because they want to try and keep science
Yeah.
unified across the board.
Yeah, of course.
Aside from the whole imperial versus metric, that was just someone
It-
tossed that out the window.
It's just the pronunciation that they say
Right.
differently, like
Yeah.
laboratory, respiratory, respiratory.
Right.
Aluminium, aluminum.
Right, exactly.
Tomato, tomato.
Mhm.
I think that's a really good point, though, when you were discussing about different names and things, especially when you're saying about Latin names. So that is one of the areas of confusion, personally for me, is when I come over here, is actually common names of taxa. So there's lots of different common names, and so that's why we always refer back to the Latin names or the true species names for different types of organisms we see on the seafloor, because what I might call a reptile, someone else might call a rabbitfish, and a scorpionfish or a batfish, and it can get very, very confusing when you're talking about the same,
Mhm.
different species or different names, and
Mhm.
it's even Ben and I came across it the other day when we were trying to think of something. I
Oh,
can't
yeah.
even remember now. Frogfish, toadfish
Oh,
versus
right,
batfish.
exactly.
Yeah, we were
Itinerary
confused
versus
as to which.
oxyphalid versus,
Yeah.
which
They're,
is actually meaningful. Versus
So it turned out you guys were both right and just talking. with
probably
your-
both equally wrong. No.
Yeah. In that point, we were both wrong.
Leah, are there manatees in Europe?
Are there
Manatees?
manatees? No, I don't believe so. Not in Europe.
More of
Oh,
a
sad.
tropical.
Hey, Lee, do you have manholes?
Well, I do think there are people whose job it is to name the fish's name like at Whole Foods based off their based off the timing and the marketing. So, what should we call a whitefish this season or a butterfish? I'm
Oh,
fairly
really?
sure. Yeah,
Whichever
there's a
one
whole.
sounds more appealing.
Yeah, my
So a butterfish from one season to the next,
maybe
depending on the local
change,
fishery, can be
right.
something different.
It's like a Chilean sea bass is a fancy name, but some years it was some deep sea monstrosity of a stomiid fish.
Yeah.
You know.
Yeah,
Well,
I
Chilean
heard
sea
that
bass
about-
is Patagonian toothfish as well, which if you look up a Patagonian toothfish, it doesn't look very pleasant
Oh, yeah.
to look at, right?
Not the most
Yeah, it's not a sea
appetizing
bass.
fish.
Yeah, not a sea bass at
Not
all.
a saranid. Yeah.
Wow, I did not know that. Huh. I always knew there were people in the supermarkets that named like fruit, and I always wanted to know if, like, who got that job. Like, these peaches are Crimson Sunset, or something fancy, but I didn't know they did the same.
I didn't even know they had names like that. I'm like Peach.
I heard that some restaurants will portray or say that they have a salmon or something on their menu, but they'll actually serve you some other fish that, you know, looks like a salmon.
That's kind of a common thing because they do that with redfish too. So
Right.
they'll serve you actual redfish and call it red snapper, which is very
Right.
vastly different.
Different.
I've heard the same thing about scallops as well. There's the sea scallops versus
Right.
the bay scallops, and one of them I think is actually shark meat.
Shark what?
Meat?
Uh-oh.
I have a quick question for Nav, for you, Ben.
Yeah.
Just really quickly, I'm looking at high pack. Target time to site 26 minutes. Is that
26
correct?
minutes. Yeah, that should be right. If Herc, are we going like the same speed?
Approximately.
More or less.
I'm going to say probably more like 35 minutes, right? By the time we stop the ship and then the vehicles swing in behind. Is that correct?
I think
Yeah,
that
it's gonna
sounds
be
fair
at least
because
30,
he's
yeah.
going to descend and
Yeah, add
we'll
another 10
we'll-
minutes there.
yeah.
Yeah, it'll be if we get there before watch change, it'll be a miracle.
A miracle. Yeah, so okay, so we're probably
500
looking at maybe
meters.
4 o'clock ship time. So yeah, 30 to
Yes.
40 minutes. So for those of you following at ISC, it'll probably take about 40 minutes for us to actually get bottom acquired at waypoint 3.
What you were just talking about.
Sorry,
so-
I'm gonna say
go ahead,
interject
go ahead.
something there. I
Sure.
think that it might be more than that. It might be more like an hour.
Wow.
The ship will get there in 27 minutes, and then there's going to be the layback of Argus, which you have to accommodate, and then the descent. So there's actually several more steps.
Oh, okay, so maybe more like an hour.
Maybe more like an hour.
Okay, copy that.
Yeah, I'd say 4:15 to 4:30 kind of thing.
Yeah, Hergus's target time now is like 50 minutes.
Well,
Okay.
that's going to be. I'm just going whatever speed. I'm
Right.
slow and then fast and then
Oh,
slow and then fast.
okay,
The best
got
one is the ship will get there in 25 minutes.
Got it.
We'll probably have 5 to 10 minutes of layback,
Wait,
5
yeah,
to 10
bring the
minutes of
layback back.
descent,
Yeah.
and then
Got it.
yeah.
So the target time in high pack, does that actually take into it? Is that using the speed and
for-
position of Hercules, or is that
It's
using?
the ship.
It's the
bold
ship.
is
Yeah,
ship.
the bold
Only
is
the-
ship.
oh,
So
right.
it's the vector from the ship.
So you see if you're watching high pack now, you'll see Hercules target time is 55 minutes. That's based on my speed over ground.
Oh, I see. Yep, got you. Sorry.
Yeah, so it's based off his current velocity. So
Sorry. Yeah, I was looking at the wrong line of data.
No,
of course. Yeah, it says now at 55 minutes.
44, 35. It's an instantaneous
Understood.
velocity. Yeah.
Oh, Trevor, you're not on SPL.
Oh no!
That's
You missed
okay.
all that big explanation.
So you did.
Guess it's gone.
So yeah, you just changed the vehicle speed just to look at the.
Just for fun.
So
So
yeah.
they
Yeah, just
calculate
for
the
fun.
Hercules target time based off the instantaneous velocity
Yeah, or
Yeah.
the smooth velocity over
of
5
short
seconds,
time interval.
not
yeah.
minutes. And whereas the ship's instantaneous or the ship's smooth velocity is actually quite smooth,
Because the ship is quite smooth and it's
Yeah.
yeah.
whereas I'm just driving to match Argus, which is dependent on a lot of things.
So we'll get there in 25 minutes, wait for Argus to catch
Settle
up,
up.
Swing in.
and then drop in.
Yep.
So a lot more time for us to talk about the funny names of fish.
Well, what I was going to say is that in New York City, some high school students did DNA barcoding on some sushi restaurants
Oh, wow.
and found
Oh.
out that it wasn't what was advertised.
right,
Yeah.
yeah,
Right.
like I was just saying.
Exactly.
Or maybe those sushi vendors also didn't know either.
Yeah.
Who knows? I don't know
It'd
where
be hard
that
to
chain
trace.
stops.
Right.
Whatever.
There
The
was
sushi
knowledge
racket.
that was not accurately represented. Who is it? Who's the culprit?
It's really interesting. I think there's a lot of work you can do now geochemically with like otoliths in fish as well. So what's their remnant ear bone, isn't it? To look at the environment they were actually living in as well. So again, if a fish was suggested it was caught in the North Atlantic, you might then look at the geochemistry of the otolith, the structure of the fish, and then you can actually see that it wasn't caught there. It was caught somewhere
I
else. So you
see.
can
False
really use these
advertising.
Just from
molecular
the
and geochemical.
precipitative environment of the
Oh, that's
calcium carbonate
interesting.
in the bone.
Yes,
Yeah,
yeah,
yeah.
absolutely. So you can really start to use these different types of tools to really investigate what's being
Though I hope I
Sold.
don't get an otolith in my sushi.
No, no one wants to know. So
You can tell how old it is.
Yeah, exactly. At least you know
Take it
how old
home
it
for
is.
some scientific experiments. Can I have a doggy bag for these tiny, tiny little things? So, you guys want to explain what otoliths are for the world?
And for me,
It's like a ship's ear bone, essentially.
Yeah.
A ship's. Still have nav on the mind, sorry. A fish is ear bone that will, cause the auditory response through its reverberation, but it's quite useful for aging fish because it kind of has these tree ring-like patterns of its deposition, so that you can get kind of annual reads to age the fish. And therefore it's useful for fisheries statistics, a lot of modeling. So you want to know what a cohort or, how the fish's ages line up versus length and all this.
When I last year, I spent a study abroad period studying animal behavior and physiology, and we got to go to the Mystic Aquarium. And
Connecticut.
we actually got to dig through some seal poop, which
Wow.
is actually super exciting. I don't know if anyone else thinks that's exciting, but we had to find the otoliths and match it to different types of fish and identify what the seal was eating based on the otoliths we were finding in the feces.
Wow.
That's dedication.
Wow.
Wow.
That's really cool.
Mm-hmm.
Yeah, only animal people will find that fun.
Biologist, right? True.
So many, many years ago, when I was doing my undergrad, I looked at brittle stars in a program called BioIce in Iceland. And what they were looking at is, we were looking at seasonal surface productivity, so the phytoplankton blooms on the surface, and if we could find any evidence of that in the deep sea. And the way we did it was using brittle star ossicles. So if you think of a brittle star arm, it's all kind of articulated, and each one of those articulations is called an ossicle. And similar to the otoliths in a fish ear, you can actually look at growth banding on these ossicles. So you can use SEM to look at a really high resolution to look at these growth bandings. So in times where there was a lot of food availability, they'd actually grow very quickly, and the porosity of the ossicles would look really big. So it looked like a sponge with really big pores. And then when it slowed down, when there was less food available, it would become really, really dense. And so you'd start to see this banding in the ossicles of a brittle star. And I remember when I was in undergrad, I was like, this is so cool to be able to tell something about what was going on in the surface waters, and it was being detected like 1,500 meters down at depth with all these deep sea creatures, and just looking at one like tiny part of it to be able to tell that was really, really cool.
Yeah, that is wild. Like Ben said with the tree rings, like you learn so much just from this one circle. And actually, they do the same thing with sea lions. The whisker on a sea lion, the farthest point out is the oldest, and as it gets closer to the face, that's the newer part of the whisker, which makes sense, but that is how they identify their biology, and they use it for mothers to understand their birthing patterns and see the hormone levels as they've grown out of the whisker. They can track the levels over the years and how they're changing.
Fascinating. So many keys nature provides to us, so much information.
Absolutely, and it takes a whole array of scientists to be able to pull all these lines of evidence together. So you'll get so many different disciplines working together on different animals to be able to kind of put all those pieces of the puzzle together to try and answer some of the big questions.
I think that's my favorite part about this kind of science is, you know, you have your one thing that you focus on, but then you realize how integral it is to someone who's in an entirely independent, well, I guess not independent, but entirely different field than you. And you guys come together and you realize you're studying the same thing, and they work together so well. So cool.
It truly is, and that's why as a biology teacher I love to spark that curiosity in the kids. They're natural scientists, really.
They are. Everyone, I always say everyone's a scientist. The moment you ask a question, you're a scientist. You want to know why or how something works, and you're going to go figure it out. And scientific process is a basic part of life. Of course, fun fact, I actually hated science fair growing up.
So getting back to fish, it's better to eat the smaller fish than the bigger fish because of the pollutants.
Yeah,
that are in it.
biomagnification.
Exactly. So that's why I never really care if I win the what is that called on the fishing boats? Oh, what do they call that when you put money in,
Lottery?
like the pool? I think it's the pool. You pool your money together,
Uh-huh.
and you put it in like $5 or $10, and then whoever wins whoever catches the largest fish wins it all. I personally don't want a large fish.
Interesting.
Mm-hmm.
I didn't know that was a thing.
Yeah, when they go out on fishing boats.
I
That's
mean, like sports fishing.
That's my excuse for not winning the pool.
Yeah, Susan, you never struck me as a sport fisherman. Do you go deep sea fishing?
With my son.
Oh.
I started him when he was 8, and he's now 31. But really, it was because he was interested.
Well, there is a concept in kind of our understanding of fisheries over time with the human population of fishing down the food web, where the first things to kind of be depleted in their population size are the larger individuals and the larger species. So it would be the larger species and then the larger individuals within that species.
Just because
So-
there's more, they need more resources.
Yeah, and I think the probabilistic aspects of catching them as well. And so you're kind of fishing down the food web where the first things to go are the apex predators, the kind of charismatic large species are more sensitive to having a stable food web supporting them sitting up top. And so, you know, there's a huge pull interest now in deep sea fisheries or mesopelagic fisheries for all these small forage fishes, this hugely abundant but small forage fishes, these bony, not good-looking fishes. And that's in part because, you know, they're now the kind of prime target on that fishing down aspect where the larger species are becoming depopulated.
Not good looking. Wow. It's pretty harsh, man.
Yeah, you know,
Mackerels.
maybe
Right.
it's
Yeah.
relative or
Beauty is in the eye of the beholder, Ben.
exactly.
Well, even what's legal now has been increased, you know, like for certain fish, calico bass, et cetera, you know, in California. And a lot of fishermen are upset about it, but they don't understand sometimes the reason behind, as you were just talking about, Ben.
Yeah, it's a hard thing to try and manage and get people to understand. Like in the early 1900s, there was the age of abundance, and everyone just assumed, here's all these animals, we can do whatever we want. And you see all those pictures of buffalo skulls that are mountains high, and trees are totally, you know, taken down, and entire forest gone, and populations of fish now. It's people think that they're around forever just because there's a lot of them, but you got to be able to manage how much of those resources you use.
Can you switch me over to USBL, please?
This is Nautilus Live off the coast of Northern California in Oregon at Gorda Ridge hydrothermal vents. We're at the base of the Juan de Fuca plate, and scientists have been exploring the hydrothermal vents over the past few days here, collecting samples so they may look at their microbiological composition as well as geological. And today the navigators and pilots just mapped the area for the geologists, and the microbiologists are reviewing their samples from the other days. Please go to nautiluslive.org expeditions to obtain more information about the subsea research.
Uh-oh, what's on one? Oh, I guess that used to be the mapping. Yeah, one used to be mapping, so it's,
Yeah, Chris just unplugged his computer, so.
yeah.
This question is for someone who has visited several dive sites over a long period of time. It says, are the effects of overfishing, pollution, and bleaching, etc., visible after repeated visits on deep sea dives?
Sorry, what was the question, Susan?
Someone wants to know if the effects of overfishing, pollution, coral bleaching, etc., are visible after repeated visits to deep sea dives. Not necessarily this site, but if you've gone on various other dives,
I would
they've
say
noticed
that those effects can be observed, but not necessarily at deep sea sites. Would you agree, Lee?
Sorry, Aubrey?
That like effects of coral bleaching and overfishing could be seen if you'd been at a site repeatedly, but not necessarily at deep sea sites.
Not for bleaching, but certainly for trawling in canyons or on seamounts. If you go over cold water coral reefs, you can see evidence of trawling, especially if we do seafloor mapping. Like when we used the ROV earlier, Chris was making a bathymetry map of the seafloor, and we spoke about side scan. We spoke about backscatter, which is looking at the texture of the seafloor. So sometimes you might be able to pick evidence up of there having been some trawling on the seafloor. But as Aubrey says quite rightly, we need to keep going back to a site. You almost need a baseline first to see what's natural and then what's happened and how long those habitats take to recover from that.
Right, and from that reference, it would be more destruction, habitat destruction of a site, not necessarily overfishing evidence based on populations.
Absolutely.
Thank you.
So we're heading over to waypoint 3 on our way to the start of the geotransits, which we're going to complete for the rest of this dive. I don't know if anybody at ISC is online, and they might be able to tell us why they chose waypoint 3. We can see from our map in HYPACK that it looks like a local topographic high. Yeah, it looks like it's shoaling, but was there any other reason as to why that site was specifically
Wait,
chosen?
shoaling or schooling?
But
Excuse me, Ben or Trevor, could you please explain to a viewer the Niskin bottles there on the left quad? They're curious about that.
So the can you hear me, Susan?
Yes, I can.
So the
Niskin.
Niskin bottles are used to collect water samples at specific depths or at a same depth, and so they can be triggered. And once they're triggered, they trap the water within that within the water column, and they can be brought up to surface while still keeping more or less their chemical composure. And so, you know, you can find the amount of dissolved oxygen in the water at that point. You know, the I don't know about density, but salinity definitely, and different, you know, characteristics of the water at that depth.
Okay.
Yeah,
Thank you,
so
Cassidy.
That's the camera on satellite 3, which I think you said is a lower left quad view. Yeah. Are we here?
Nearly.
So close.
So close, but so far away. I don't remember what that song is.
Now I'm trying to think of it.
Oh well.
Come up some point. Probably at an inconvenient time where a lot's happening.
Should I winch up? Okay.
So, Dr. Marsh, could you please share about your background, please?
1 second.
Okay.
Cassidy, I'm like really trying to think of it now. You're driving me nuts.
So I can give you a really quick background, Susan, as to how I got on board the Nautilus. So I actually joined in 2013. There was a UK-US collaboration to work on the Cayman hydrothermal vent fields in the Caribbean. And at the time I was studying for my PhD, and I was actually focusing on hydrothermal vents on the East Coast ridge in the Antarctic. But my supervisor at the time, who was based at the University of Southampton, Dr. John Copley, was part of that collaboration, of which Chris German, who is actually at the ISC, was part of as well. And because he wasn't able to make it on board, he actually sent me instead, which was fantastic. So that was my first experience of the Nautilus in 2013 as a scientist. And then from then on, I've been coming back every year as either a data logger or as a watch leader, helping out for a few weeks every season on board, and usually as part of deep sea ecology legs, so whether they be hydrothermal blends or seamounts or anything like that.
Fantastic.
Yeah.
Thank you.
Hey Susan, I have a question for you. Can you explain your role aboard the Nautilus?
My role
Yeah.
as a science communication fellow.
Yeah, what's your job?
My job is to communicate to the public what is happening, going on inside the control van, and also to have some interactions that teachers, classrooms, museums can actually request 20 to 30 minutes of time on live interaction, and then we can have a scientist come aboard, well, join us in the live interaction, and we will answer your questions directly with students or at your museum. And so I will take this back to my school and also develop some curriculum to share with other teachers that will also be shared on OET.
Yeah.
.org/STEM. And just learn about the dynamics here. I know that what I'm going to do is create a little mini team like this team of scientists where students will have to actually use little robots to manipulate go to goals, and everybody will have a specific job, just learning to contribute and be part of the whole research team. So yeah, it's been a great experience.
So is it a difficult process for museums or classrooms to request, time communicating with us aboard the ship?
No, not at all. They can go at nautiluslive.org, and there is a link there to request an interaction. And then they contact Kelly Moran, and she will give them a specific time. And we'll even get up early in the morning if it's someplace globally on the other side of the world that to accommodate their schedule.
Yeah.
No, it's not difficult at all. In fact, thank you for asking that, because we encourage more clubs, churches, museums, groups, not just schools, to become more involved with Nautilus Live. It's a great opportunity for people to see the interaction of scientists and experience authentic science and just learn. It's a dynamic group of people here.
That's super cool too, that you are going to bring this into your classroom and let your students, experience what we're doing here in their own way. I think that'll be really exciting for them.
We're supposed to be teaching students how to collaborate and work as a team, and this is a perfect example.
Absolutely. I think there have been a couple situations too where we laugh at how well we communicate by our lack of proper word usage, and then we just figure out what the other person means.
Semantics is everything in life.
Like Trevor was saying earlier, as long as you can work well with the team, you've got a good chance of being able to come on board here.
Attitude is key.
Absolutely, all throughout life.
All throughout life.
So for the navigator and pilots, did you have to learn how to write code? Do you have to write your own programs for the ROVs?
No, the role here is mostly operational. So for our own kind of like my own personal project in graduate school, I do a lot of coding, but it's useful to understand how these software may be derived, and by the logic and by the method, but it's not necessary at all.
Looks like we're eventually going to go north too, because you want that guy there kind of thing, I think. Sure, yeah, that's fine. Yeah, okay. Sure, yeah. Right. Underdamped. Overdamped.
I encourage you to follow us on social media, whether you're on Instagram, Facebook, Twitter, or go to the YouTube channel as well to look at Nautilus Live, and of course nautiluslive.org on the web. Please share nautiluslive.org with your schools and communities so that we can really see the application of STEM and STEAM occurring in real time aboard the Nautilus. The exploration. The exploration of hydrothermal vents may give us clues into our own history as far as the evolution of the Earth, as well as within our solar system. So it's critical that we study these particular areas, especially where there are extremophiles, organisms that live in extreme conditions. When we say extreme conditions, we're referring to high temperatures that most organisms would not be able to withstand, high pH, not even being able to photosynthesize. They use chemosynthesis instead. Dr. Robert Ballard was the first to discover the hydrothermal vents off of the Galapagos in 1977, and that was very revolutionary in biological science. Prior to that, everyone thought that photosynthesis was the only means of obtaining energy and from the sun. And now we know deep beneath the ocean, where sunlight cannot even penetrate, organisms can survive using chemosynthesis. So I encourage you to go to nautiluslive.org, expeditions, subsea, to learn more about this particular expedition. Thank you. Presently, we are changing watch, and that is what you're hearing. We are not done for the day. we will be continuing Nautilus Live, but usually the next watch will come on from 4 to 8. The 12 to 4 group will be leaving.
Well, hello, 4 to 8.
Hello.
Hi, Wayne. I guess it's just us.
Yeah, we're all alone.
Yep. Well, for the folks watching and wondering what's going on right now, we are just doing a watch change. So you're going to hear some new voices come on. Voices from the 12 to 4 shift get to head out of the van and have a little break before dinner. So we are actually in the middle of our 5th dive of the Gorda Ridge expedition as part of the Subsea Research Program, which is a collaboration between NOAA, NASA, Woods Hole, Oceanographic Institution, Ocean Exploration Trust, and a whole plethora of academic research centers across the country. So if you're curious to learn more about this expedition on the whole, I'd encourage you to check out the expedition page on Nautilus Live. Meet some of the team on board, see who's talking to you right now and whose voices you'll hear, and get some more background on that program. And keep sending in questions. We are gotten a little bit of an update, but it sounds like we are in transit to our next site where we'll do some more visual surveys and collecting samples. Is there anyone in the science row who could speak a little more to our goals for the rest of this dive?
And please send in questions, because otherwise Sam makes up questions.
Who would tell you that?
She's been found out.
Ship rumor.
Someone out there in the world is wondering these things.
Ship rumor.
Hello, we just did a watch change. Darlene just put on her headset. It sounds like somebody would like to talk.
Hello, Darlene, who just put on her headset.
What's happening?
We are just getting folks caught up with what we're doing. A lot of folks tuning in wondering, where are we going? Are we done for the day? And we are definitely not. This dive is actually going all the way until 4 a.m. Pacific time. So we're doing 24-hour operations today. So Darlene, we were just hoping to get a quick overview of kind of where we're at for the next 4 hours of our watch shift from 4 to 8,
okay,
and what viewers might expect.
So what they can expect to see is actually a lot of blue water for now while we transit to what's called Waypoint 3. Waypoint 3 was actually not put into the plan, but it was supposed to be there, so we had to drop it in. And we don't actually know what we're supposed to do when we get there. I think that the ISC is on dinner break. And so we're just waiting for them to come back and tell us what to do when we get to Waypoint 3.
Coming down.
From Waypoint 3, we're going to actually
Shannon,
head out.
can you hear me?
Hey, Shannon.
Hey, we finished eating and we all got back.
Great. Do you want to tell us what we're doing at Waypoint 3, please?
Absolutely. So there is a mound there that is noticeably tall. It's not really connected to any of the regular scarps. We're wondering if there's a chance that that is a hydrothermally formed mound that could have been caused by seeps. So we want to blue water over, drop down, take a look at it. If it has seeps, that will change what we do. Then if it has nothing there and is just a rock.
Okie doke. Sounds good. So you guys can direct us once we get there. Miles, what's our distance to target, please?
11 meters.
Cool. So we're almost there, Shannon.
Yep. We'll
plus
descend.
plus 150 vertical.
Okay, so we're not almost there. We're almost there.
Minor details.
I mean, as the sea crow flies, we're pretty much there.
So we've had like 3 fin whales playing around our boat all day since early this morning. And my fingers and my toes are crossed that they'll be there when Herc comes back up. Because that would be epic.
That would be epic. Got a little bit of time to fill, though, before we get there.
Yeah. And so, Samantha, after that, we're going to head off to 6 different geotransects, or geotransect waypoints. And what a geotransect is a mapping transect, a visual mapping transect for the geology team at ISU, Idaho State University. And that's going to proceed for about 13 hours. So that's going to be what the bulk of the remaining time of this dive is allocated to, are those geotransects. And so that'll come after Waypoint 3.
Excellent. And what's involved in a geotransect?
We're going to basically look at the bottom, have the camera pointed down with the lasers on. Lasers are 10 centimeters apart. So that gives a relative scale for our geologists to make inferences about what they're seeing. And the geologists are going to be making notes, virtual notes or e-notes, if you will, about what they're seeing. And they're going to look at changes in the rock and the rock shape, the morphology of the rock. They're going to look at how much sediment is there, how it actually kind of interweaves itself between the harder rock types and so forth. All of that information will get compiled and then sort of reconciled with some of the rock samples and the data that'll come out of there eventually. So there's a lot of components that make up the geology research element of subsea. It's pretty complicated.
And in addition to the team we have on board, the science team we have on board, there's also a team back at the Inner Space Center at the University of Rhode Island Graduate School of Oceanography, right?
That's right. I don't know if they want to...
What's our bearing to target?
10.
All right, so we'll have to turn north.